Hey everybody, it's Mr. Bliss. I am making a video today from school, so audio might be a little bit off, but that's okay. Today's video is about nucleic acids. It's the fourth video on macromolecules, the last one that we need to cover. And it's going to be our gateway into talking about genetics and later things in biology. It's pretty important. So our story here actually starts way back in 1866. With a Czech monk. He's an Augustine monk named Gregor Mendel. And Mendel was an avid gardener. He would spend a lot of time in his garden looking at the variety of the plants in his garden. Specifically, he looked at the variety of plants in his pea plants. He noticed that some of them grew tall, some of them were short, some of them had white flowers or pink or purple flowers, and he was wondering if he could manipulate these in a particular way. So what he did was he cross-pollinated certain varieties, kept track of this, and he was the first one to identify the concepts of dominant and recessive traits, what we call heredity. And so this was the first scientific investigation into how traits were passed on. He had no idea what controlled this, and in fact, we didn't know for another 100 years, really, the mechanisms behind these things. And we're still figuring them out today. After Mendel was a Swiss psychological chemist, and his name was Friedrich Miescher. And Miescher actually looked into the proteins and things inside of discarded bandages, which is pretty gross, actually. He looked at pus, for example, in an infection, and he realized that there was a new substance in there, and there was a substance that he called nuclein, because he knew that it originated within the nucleus of cells. And so here's Friedrich Miescher checking out some bandages on his microscope here. But he knew it was unique. After that, we unfortunately stumbled into some dark times within the world of genetics. In the early 1900s, up until the 1940s, there was this idea of eugenics, the idea that you could perfect genetics and have some dominant races, for example. And this led to all kinds of problems, scientific racism. Unfortunately, it was heavily politicized and involved in, for example, Nazi-era Germany. And this is an era that we refer to as science-led astray. And we still use it as kind of a really important moment where we realized how important it is to employ ethics within biology and to make sure that we're doing things in the right way, that we're not disenfranchising whole groups of people, and doing things for the sake of all humanity. So much later on, 1944, there was a Canadian-American biologist. His name was Oswald Avery. And what Avery did was he looked at Miescher's studies with nuclein, and he started manipulating them. And what he did was he took this substance that we had now identified as being DNA, and he transferred DNA from an inert species of bacteria that causes pneumonia. And, he transferred it from an active species of bacteria and transferred it into an inert species. And what happened was the functionality of these bacteria changed. It became a virile strain again. It could cause pneumonia. And he referred to DNA as the transforming principle. This was the first moment where we definitively knew that DNA was what carried genetic information. Previous to this, we suspected that it was in the proteins within cells, but now we know that this Miescher's nuclein, this DNA molecule, carried genetic information. So let's talk a little bit about nucleic acids. The function of nucleic acids, there are two. One is the storage of genetic information. We are going to see that in a molecule known as DNA. And then also the transfer of that genetic information. So the information that's stored that makes you you. So just like all of these other macromolecules, nucleic acids have a monomer form and a polymer form. This here is the monomer form of a nucleic acid. It is called a nucleotide. And nucleotides are made up of three parts. The first is a phosphate group. So here's our phosphate group over here. It's the same phosphate group that you see in phospholipids. Then we have a 5-carbon sugar. It's a carbohydrate. And there are two 5-carbon sugars that help to define which nucleic acid we're looking at. You can either have ribose, which is found in RNA, or you can have deoxyribose, which you find in DNA. And the last aspect, the last component of a nucleotide is a nitrogenous base or nitrogen base. And so it's made of nitrogen and other components. And the structure of this nitrogenous base actually tells you which base you have. You can have one of 4 in DNA, either adenine, thymine, cytosine, I wrote thymine twice, or guanine. And we just refer to these by their first letters, A, T, C, or G. This one here we will call adenine. So that's the monomer form. And then the polymer forms are like this. You have deoxyribonucleic acid, or DNA, and then you also have ribonucleic acid, or RNA. And here are some differences between them. DNA, or deoxyribonucleic acid, is double-stranded. This is DNA here. It forms what we call a double helix. With our sugars and phosphates in this backbone chain right here, and our nitrogenous bases forming kind of like the rungs of this DNA ladder across the middle. By contrast, ribonucleic acid is single-stranded. It has ribose sugar instead of deoxyribose. And instead of thymine, we have a nitrogenous base known as uracil. So instead of A, T, C, and G, we have A, U, C, and G. So if we were to zoom in on DNA here, okay, it would look a little bit like this. So here is one nucleotide, and you'll see that it kind of forms a chain like this, and these all point in this direction. And there's a second chain that forms pointing in this direction. And your nitrogenous bases line up in the center here. So these bonds that hold the backbone together are very strong, but the bonds that hold these bases together are actually fairly weak, and they can break, which is important for processes within genetics. The other thing that you'll notice here is that there was a discovery in the 1950s by a man named Erwin Chargaff, and Chargaff came up with a set of rules. He sequenced some DNA, or not sequenced, but he measured the amount of different chemicals in the DNA, and he came to the conclusion that the amount of adenine that was in a section of DNA is always equal to the amount of thymine, and that the amount of guanine is always equal to the amount of cytosine. And so he postulated that these paired up with one another, and he was correct. So adenine always pairs with thymine, and cytosine always pairs with guanine. And there's a mnemonic for this. The way that we remember it is that apples are in the tree, A, T, and the car is in the garage, so C and G. And I believe that's a great mnemonic that came from the Amoeba Sisters videos, and I will link those in the description. The other thing that Chargaff discovered is that DNA is species specific. So the amount of DNA that you could find in humans is similar between humans, and the amount of DNA that you can find within monkeys, for example, a species of monkey, let's say rhesus monkeys, those are similar between rhesus monkeys, but they're different between species. So he postulated that DNA helps to define that species, and that was really important. Chargaff didn't know the actual structure of DNA molecules. That came later, but he was able to use this information and synthesize this information, and other people use Chargaff's rules in order to discover that sequence and that structure. Here are some of those people. So in 1952, a couple of years after Chargaff, there was a woman named Rosalind Franklin. And Rosalind Franklin's work was vital for understanding the structure of DNA. She did this technique known as X-ray crystallography, where she took images of very small things. And she has this famous photo known as Photo 51. She was working at King's College in London, and unbeknownst to her, her photo was shared and viewed by some other researchers in her building, specifically James Watson and Francis Crick, who have been credited with the actual discovery of the structure of DNA. Unfortunately, because of her work with X-rays, Rosalind Franklin developed ovarian cancer and died at a very early age. And so she was never honored the way that she should have been, which is, getting the Nobel Prize that Watson and Crick did get. So this is James Watson and Francis Crick. One is an American, one was Brit. And they were the first ones to publish a relatively short paper that outlined this structure of DNA. Since then, there have been lots and lots and lots of discoveries with DNA, the entire field of genetics. We learned about the links to certain diseases like Down syndrome, trisomy 21. We later learned about protein synthesis, how to sequence DNA. In the 1990s, we started the Human Genome Project, mapping DNA sequences with the use of computers. In 1996, we cloned the first mammal, Dolly the sheep, which was massively important. And then even now, we're discovering, really important techniques like CRISPR-Cas9, which is a gene editing program. It's fascinating. Later topics in this course, we're going to be talking about DNA replication in our next video. And then we will eventually talk about protein synthesis, transcription, and translation, which is the process of taking information that's stored on DNA, how it's transferred to RNA, and how that's used to build proteins. And proteins, as we know, make us and the characteristics that define us. Anyway, that is nucleic acids. I hope that you found it useful. Watch it again, ask questions, and I will try to answer them in the comments. Thanks.